wonderful to get the seminar series back going again. We've missed it, I think. I'm sure we have. I certainly have. I haven't missed chairing them, but I've certainly missed them. So we're kicking this session off, and there'll be seminars likely each couple of weeks, and they're going to rotate through ag and environmental science and ag resource economics. So it's actually going to be quite exciting, I think, and it would be wonderful if people don't just stick to their own little favourite pet topic and come and hear more generally what's going on in the world of the School of Ag and Environment. And we're planning to get industry people engaged in the series as well. And so that's a good chance to network and hear other things that are going on in the state and in different institutions. So please support the seminar series. It's a friendly place for critical assessment of what you're doing and getting that really useful feedback about whether you're communicating your information clearly through to an audience. Jiayin Pang first, talking about improving phosphorus use efficiency of chickpea. And so that will be 20 minutes, and we'll have some time for questions. And then we'll have Gerelee speaking on the tolerance to waterlogging and recovery ability among the 3 subspecies of subterranean clover. And similarly, we'll have a similar length of time and then questions right at the end. And then we'll wrap it up at 1 o'clock. So if I can just invite Jiayin to start us off. Thank you very much for starting our series. I'm sure you volunteered, and we need plenty of those, so thanks very much.
Good afternoon, everyone. Thanks for coming. Today, I'm going to talk about improving phosphorus use efficiency in chickpea. Chickpea is a self-pollinated diploid food legume. It was originated from southeastern Turkey, later was spread to India and Europe. Now it has been grown in over 50 countries around the world. It is ranked second among pulses for world production. There are mainly 2 types of chickpea, daisy and kabuli. The one on the left is daisy, the one on the right is kabuli. As you can see, the kabuli seeds always have darker colored seed coat. Seeds are smaller and seeds often look a little bit shriveled. But kabuli seeds, they are much bigger compared with daisy, and seeds are light colored. It's either white or cream colored, and seeds are smoother, and the seed is much bigger, and probably the cost is a little bit more than the daisy. All these food, I didn't cook them, but I grabbed them to make you feel very attractive. All these food actually made of chickpea. They are very nutritious. They contain high iron. Also, chickpea provide good source of fiber and minerals. As a food legume, the most important function in agriculture system is it can fix up to 140 kilogram nitrogen per hectare. So therefore, it not only provides itself for enough nitrogen, but also leaves a large quantity of nitrogen for the subsequent crops. So it is important part of for the sustainability of agriculture ecosystem. For the world chickpea production, India is the largest chickpea producer. For example, in 2017, the world chickpea production is 14.6 million hectares. India is 9.6 million hectares. About 70% of chickpea is actually produced in India. Then followed by Pakistan and Australia. In recent years, so for example, in 2017, Australia has overtaken that in Pakistan. So Australia has become the world's second largest chickpea producer. Australia is chickpea production in Australia is relatively new industry. It started in late 1970s. By the end of last century, it reached a reasonable area about 300,000 hectares. But because of the outbreak of a devastating fungal disease, it's called Ascochyde blight, the industry has set back quite a lot. But at that time, Australian chickpea breeders have worked very hard to breed some Ascochyde blight resistant chickpea cultivars. So starting from 2005, the industry come back steadily. Until 2017, it reached over 1.1 million hectares production area. It becomes Australia's #1 grain legume. So mainly it's produced in Queensland, northern part of New South Wales, some in southern Australia, including Victoria, South Australia, and some in Western Australia. However, during the process of domestication, a lot of genetic diversity have been lost. So one of the problems with the current commercial cultivars, they all have quite a narrow genetic base. To overcome that problem, 𝒂𝒂𝒍, which is based in India, in recent years has developed a unique chickpea reference set, which has 300 genotypes, which were originated from 29 countries with diverse genetic background. Phosphorus is essential for all lives on the earth, including animals, plants, fungi, bacteria, and even in viruses. It is a very important structural component for the cell membrane and nucleic acid. However, like fossil fuels, rock phosphate is limited. It's very limited, and also it's non-renewable. Rock phosphate mainly distribute in 3 areas around the world. The first area is Morocco and Western Sahara, which holds about 45% of world phosphorus reserve, then followed by China and the United States. But even if we add up all the global phosphorus reserves, it is estimated that the whole reserve could only be enough for the next 300 to 400 years. The phosphorus cycling in the ecosystem always starts from the production of phosphorus fertilizer from rock phosphate deposits. When phosphorus fertilizer is applied to the field, a small proportion of soluble phosphate will be directly taken up by plants used for the agricultural production. A very large proportion of phosphorus will be fixed by soil particles, and so that part of phosphorus will be locked away. For example, in acidic soil, phosphorus will be locked away by oxides, hydroxides, aluminium, and iron, and in alkaline soil will be locked away by calcium. So that part of phosphorus is not available for most of plants. When the crops are harvested or any animal products were taken away from the paddock, that part of phosphorus will be exported to the food system. But meanwhile, any crop residues and the manures, when they return to the field, they become the organic phosphorus source. Of course, between those different phosphorus forms, they will have some transformation between each other. In the cropland area, often the phosphorus fertilizer input and the phosphorus removed from the field is imbalanced. If the phosphorus fertilizer input is less than phosphorus removed, that can cause the issue of P deficit. But on the other way around, if the phosphorus fertilizer input exceeds the phosphorus removed, that can have the phosphorus accumulation in the field, cause the problem of phosphorus surpluses. In a very large area, for example, in most part of Europe, in Southern Asia, and most part of North America, and a large part of South American a lot of field paddocks already got over-accumulated phosphorus. So the question is, the total phosphorus is building up in the field, but does that mean also the plant available phosphorus also increasing accordingly? We have some data from a paddock along the southeast coast China. We found that total phosphorus accumulated to very high, up to 600 ppm. But if you look at plant available phosphorus, it's relatively low. It's only like 10, which is not enough to have the high yield. Therefore, there is large gap between the total phosphorus and plant available phosphorus. Therefore, it would be very important to increase phosphorus use efficiency. The first pathway would be to improve phosphorus acquisition from the field. To achieve that, we need to have better genotypes through breeding programs. And also we need to, through the appropriate phosphorus fertilizer management, we need to organize phosphorus input and improving 𝒂𝒃 utilization. 𝒂𝒃 means the phosphorus being locked in the field. And also we need better research on below-ground traits associated with improved phosphorus acquisition. The second pathway would be to improve the phosphorus utilization efficiency. Once plants take up all those phosphorus, how plants can produce more biomass or more yield. Instead of, for example, for a certain amount of phosphorus, it produces 2 grams or 10 grams. That can make a big difference. So in response to low phosphorus, plants have evolved a number of below-ground traits to improve phosphorus acquisition. For example, plants can allocate more proportion of carbon source into the roots to increase root mass ratio. And plants also change the root morphological traits, for example, root diameter, basic root length, and many other things to make phosphorus acquisition more efficient. And also plants often form mycorrhizal association that would increase the volume of soil explored beyond the root depletion zone. And often plants exude a large quantity of exudates. For example, this is a diagram by Hans Lambers. For example, if plants exude carboxylates, as I've just mentioned, a lot of phosphorus were locked up by soil particles. That carboxylate can take the place of either inorganic phosphate or organic phosphate. It can push those part of inorganic phosphorus and organic phosphorus into the soil solution. If this organic phosphorus is further broken down by enzyme, it can become inorganic phosphorus that can be used by plants. So in our program, we included over 250 chickpea genotypes from chickpea reference SAgE, plus some Australian commercial cultivars. And plants will grow under extremely low phosphorus. And there are large variation in genotypes in plant growth. For example, some genotypes, this is only like 3 weeks after planting. At the bottom leaflets, some genotypes show the necrosis or the purple colored. And over the time, these bottom leaves even getting more yellow, and finally they were dropped over the time. But in some other genotypes, there is no symptoms at all. So for the plant growth, there is large variation among all those genotypes. For example, for the shoot driveway, it varied from less than 0.1 to about 0.6. There is like 6 times variation. And similarly for the root dry weight, root mass ratio, and both shoot phosphorus concentration and shoot P content also showed very large variation. Physiological P use efficiency and P utilization efficiency, both of them are actually the dry mass they could produce given a certain amount of phosphorus. Again, there are large variation among those genotypes. And both area-based photosynthesis and mass-based photosynthesis also got large variation. And leaf transpiration rate and water use efficiency varied very significantly among different genotypes. And photosynthetic phosphorus use efficiency also varied. And that's the root morphology data, including total root length, root surface area, mean root diameter, specific root length, and root hair length. They all got large variations. And this is rhizal sheath size. Here I maybe explain a bit what is rhizal sheath. If we take plants out of the pods, we have a bit of shake. Any soil remaining attached on the soil, on the roots, are called rhizal sheath soil. So there is large variation in rhizal sheath soil dry weight and specific rhizal sheath soil weight. We found rhizal sheath soil pH was acidified by root oxidates, and again, large variation among genotypes. And for the carboxylate, there is large variation among genotypes. It ranges from 20 to 120. There is about 6 times variation. And here I would like to point for chickpea, the predominant carboxylates are actually malonate. It occupies 50 to 80% of carboxylates, then followed by acetate, citrate, and malate. And mainly it's malate for chickpea. We made all the correlation among all those parameters we measured. We found that total root length and root hair cylinder volume had very strong correlation with shoot P content. That shoot hair length is a function of mean root diameter, that's root hair length, and total root length. And also we found carboxylates had a very strong correlation with phosphorus acquisition, particularly malonate. And very interestingly, we found that leaf transpiration rate had good correlation with leaf phosphorus concentration. During the daytime, when leaf stomata are open, so water moves from the soil upwards to the canopy. That's a process called a mass flow. During that process, with the movement of water, some minerals can also move to the root surface. But in soil solution, often the inorganic phosphorus concentration is rather low, so it is generally assumed that the role of transpiration-driven mass flow in phosphorus acquisition is rather minor, but in our case, that's not true. So here I would like a little summary of this section. So for the chickpea phosphorus acquisition, leaf transpiration-driven mass flow is important, and so some root morphological parameters, including total root length, root surface area, and root hair cylinder volume. And also root carboxylates, particularly malonate, are important for the phosphorus acquisition. As I have just mentioned that a number of processes, below ground processes, are important for the phosphorus acquisition. But all those below-ground processes, they are actually carbon costs. They need some investment. So the question here is, how would all these below-ground traits be coordinated with each other to acquire phosphorus? So to answer that question, we selected 10 chickpea genotypes with high carboxylates here on the right side, 10 genotypes with low carboxylates. So you can see the genotypes with high carboxylates often had taller plants compared with genotypes with low carboxylates. And that's extremely low P, that's moderate low P. Under both low P environment, those genotypes with high carboxylates, they had less branches compared with here. But under high phosphorus, there is no that difference. The genotype with high carb also got plenty of branches. And we looked at the root morphology data. We found here, this one, the gray one, is genotype with low carboxylates. The white one is high carboxylate. All the genotype with high carboxylates had lower root diameter, which means roots are thinner, and low root mass density, but high specific root length and high specific root surface area. And very interestingly, under both low phosphorus conditions, that high genotypes with high carboxylates also had high AMF colonization here and here. So here we have a little summary. For any genotypes with high carboxylates, they also have high AMF colonization, but thinner roots, low root mass density, low root mass fraction, but high specific root length. As I have just mentioned, carboxylates are important for the phosphorus acquisition, but to measure that is quite tedious and labor intensive. So if we can find anything more simple, any like more easily measurable proxy for that, it will be useful for the breeders. So again, I read back to this diagram. As I just mentioned, carboxylates could mobilize fixed phosphorus from soil particles, but also carboxylates can mobilize some cations, for example, iron, manganese, zinc, and copper, make them available and being taken up by plants. So we verified that theory in chickpea. We found very strong correlation between leaf manganese concentration and below ground carboxylates, particularly with melanate. There is strong correlation. That's among 100 chickpea genotypes. And again, we found strong correlation between leaf iron concentration and carboxylates. Zinc as well, but the correlation is a bit weak compared with manganese and iron. And we further verified that theory on the 3 different P levels using those 20 genotypes with contrasting carboxylates. We found again on all P levels strong correlation between manganese and carboxylates. And iron also have very good correlation with melanate on all 3 P levels. But for zinc, strong correlation on the low phosphorus, but not on high phosphorus. So we found mature leaf, zinc, manganese, and iron concentration can be used as easily measurable proxies for the assessment of carboxylates and the low phosphorus environment. That would provide valuable information for the breeders and selecting genotypes with high phosphorus acquisition efficiency. So this is my last slide, take-home message. Firstly, I would like to say that chickpea collection provides very useful genetic diversity. It can be used as very valuable source to improve phosphorus use efficiency. And there are carbon trade-offs among the below-ground traits. For example, genotypes with more carboxylates often had high MF colonization, thinner roots, high specific root length, low root mass ratio, and low mass density. And leaf, manganese, iron, and zinc could be useful for the breeding programs, and that can also be applied in other crops and legumes for the breeding programs. I think I missed one slide at the beginning. It's the most important one. All the work I presented is our team effort. Particularly, I would like to acknowledge three important people, Siddiq and Megan and Hans, for your excellent mentoring and supervising of the program. Thank you.
Thanks very much, Jade. Now, there's a prize for asking the first question in the new seminar series. Any questions? Kevin, you get to chair the next session. No.
Movie prize. Excellent presentation.
Thank you.
Just a quick question. Given that chickpea, the end product is the seed, you've gone to the point you're still at a vegetative stage in your looking at the acquisition changes. Have you given any thought to going into the seed yield component, looking at what happens with the amount of P on board the seed as well as the proxy?
Yes. In our new linkage project, that will be one of our aims to actually lower the seed phosphorus concentration. Also, seed phytate, mainly it's as phytate. Phytate is too much of phytate is actually not good for us. So our aim would be to screening any genotypes with low phytate. That means also maybe accompanied with that process, the less phytate, we expect that more micronutrients, for example, zinc, iron, those micronutrients might be available for our health benefits.
Perfect, thank you.
Other questions? No time for the second question.
It's apparently carboxylate is very important for all those elements you studied for their availability. I wonder whether it's possible to apply carboxylate acid as a type of fertilizer.
Carboxylate, it's basically it's carbon. It provides carbon source for the microbe. Once you apply it into the soil, like exogenous application of carboxylates, it can be degraded by microbia very quickly in soil because they provide carbon source for those microbia. You might get better microbial diversity in soil that can indirectly benefit plant growth. Not necessarily the carboxylates will be lasting there forever. That's my guess.
I think the question is whether the external application of carboxylate, like malic acid or something, will solubilize the soil-bound phosphorus or not.
I say just it's easy to be degraded by microbe, not necessarily being there forever to play that role. But because plants are there, plants are alive, they can keep exuding. I think that's kind of different from exogenous application. I don't know.
There was one more hand up, but just a quick question. Was there a question?
Online?
Oh.
We are live now.
Sorry, live people.
Hans question, why do you think the correlation with leaf zinc does not persist at high pH, whereas that with leaf manganese and iron does?
Can we discuss that later?
That will be discussed later.
Hans knows the answer.
Just discuss that later over a cup of coffee, huh?
He's on a red wine.
Yeah, that's right. I think we'll have to leave it there. Can you ask the question later on, maybe? Yeah, thanks very much. And look, thanks very much. It's very naughty of you to put you.
Hello everyone. So I'm going today talk my key results of my experiment that I explored tolerance to waterlogging and recovery ability among the 3 subspecies of subterranean clover. I would like to acknowledge the funding organization. So this project is as part of the annual League on Breeding Australia, joint venture between PGG Rice and Seeds and UWA. And this project is under the international collaborative program between Nagoya University and UWA. So I am supported by Australian Government Research Training Program Scholarship and Science Industry PhD Fellowship Program. So waterlogging is a common abiotic stress that can cause significant loss on crop production worldwide. And in southern Australia, waterlogging covers large farmland area that has been estimated 2 million hectares in Western Australia and 4 million hectares in Victoria. In Mediterranean climate regions, annual plants can suffer waterlogging from excess rainfall during the growing seasons. And also Mediterranean climate regions are predicting more variable growing seasons, with increasing both intensity and frequency of the heavy rainfall events, such as a result of climate change. In southern Australia, waterlogging occurs in the poorly drained duplex soil, which is estimated 40 to 60% of the total agricultural land area. So there's estimated cost of $90 million cost per annum in terms of the crop production loss because of the waterlogging. So therefore now challenges needs to improve persistence and resistance of pasture plant in order to maintain the productivity. Subterranean clover is the most successful pasture legume in southern Australia because of its well adaptation to the Mediterranean type climate and inherently low nitrogen soil. So it contains several attributes such as range of cultivars with different maturity, enables them growing successful in different environment with a range of growing season lengths. And it is simply managed, such as ability during bursts, resistance under heavy grazing, and tolerance to many herbicides. And also widespread farmer experience throughout the historical usage makes subterranean clover the most important pasture legume among the farmers in the foreseeable future. Subterranean clover consists of 3 diverse subspecies, subspecies Subterraneum, Brachycalypum, and Jiayin, each adapted in different soil types. So among them, subspecies Jiayin is quite rare in the wild, comprises only 2% of total 𝒂𝒊𝒄𝒖𝒍𝒕𝒖𝒓𝒂𝒍 genotypes. And former studies have been found that the subspecies Jiayin generally exhibits higher waterlogging tolerance than the 2 other 𝒂𝒊𝒄𝒖𝒍𝒕𝒖𝒓𝒂𝒍 subspecies. Therefore, cultivars of subspecies Jiayin are commercially important and broadly used in high and medium rainfall areas where the most animal production occurs. However, there are conflicting results and insufficient evidences to ascribe tolerance. However, there are conflicting results and insufficient evidences to ascribe waterlogging tolerance difference among the 𝒂𝒊𝒄𝒖𝒍𝒕 subspecies. And also former studies only focused on morphological and anatomical responses under the waterlogged condition. And also recovery stage after the waterlogging stress has often been overlooked and studied little. So recovery is especially crucial for the regenerating annual legumes, for the seed set and seed production, and recovery stage hasn't been studied among the subflower genotypes. So therefore, in this study, I aimed to explore tolerance to waterlogging and recovery ability among the 3 subflower subspecies in order to guide future subflower breeding activities. We selected 9 cultivars of subflower, 3 cultivars from each subspecies. So these cultivars were selected because of the usage in former waterlogging experiments and the range of the maturity, and also usage for the parents, as a parents for other cultivars. I used also cultivar frontier. So even the frontier is a different species, the subflower, it's well known of its high tolerance. So therefore I used cultivar frontier as a check species. Experiment had a randomized plot designed with 4 replicates. Seeds were germinated in the petri dishes, and we used 4.5 liters free draining pots and mix of soil, river sand, and loam. Soil was inoculated by appropriate rhizobium and applied essential nutrients. This experiment conducted in the temperature-controlled glasshouse at UWA. Plants were grown for 28 days under the free draining condition by watering 80% of field capacity. Each pot contained 3 plants, and pots were experimental unit. There were two treatments after 28 days of growth and prolonged for 35 days. Free draining control treatment, which is plants were growing under the free draining condition for the entire duration of this experiment. Waterlogging treatment, pots were inserted in non-draining pots, non-draining buckets with similar external diameter, and water level was kept 10 millimeters above the soil surface. After the 35 days of waterlogging, waterlogged pots were drained, and plants were growing under the free draining condition for 21 days under the subsequent recovery stage. Before I start to talk my results section, I would like to explain my graphs here. So each graph panel contains the 2-way ANOVA results run between the subspecies and treatment effect and the interaction between subspecies and treatment. So cultivar frontier excluded from the ANOVA analysis because it's different species. The results are made of the 4 replicates. White bars are indicating control, blue bars are waterlogging. Black solid lines are estimated means of the control, and red dashed lines are estimated means of the waterlogging. So in this experiment, tolerance to waterlogging and subsequent recovery were defined as the maintenance of the shoot growth or the accumulation of shoot dry bed relative to the non-waterlogged condition. The results showed high variation among the subflower subspecies with superior tolerance in subspecies 𝑨𝒈𝒄𝒂𝒍. So waterlogging had a less effect on both shoot growth and shoot dry bed in subspecies 𝑨𝒈𝒄𝒂𝒍 compared than the 2 other subflower subspecies. Also, waterlogged varied among the cultivars within subspecies. For example, cultivar Denmark showed the significant high waterlogging tolerance compared than the 2 other cultivars from subspecies Saturnium, Mount Barker, and Sydney Park. So the performance of cultivar Denmark was under the waterlogged condition was almost similar with cultivars of subspecies 𝑨𝒈𝒄𝒂𝒍. So this suggests that the future study should focus on evaluation of intraspecies variation in waterlogging tolerance. And the cultivars of subflower showing higher waterlogging tolerance, like cultivar Denmark and cultivars of subspecies 𝑨𝒈𝒄𝒂𝒍, almost reached to the shoot growth as the well-drained condition, while cultivar frontier growth was beyond of its control. So from here we could summarize that the genotypes of subflora with high waterlogging tolerance has the ability to overcome transient waterlogging. So in subsequent slides, I would like to discuss more details of the main key traits and physiological responses contributing to this water-flow intolerance. So the picture here shows the root growth under the water-flow condition and the well-drained condition. So severe reduction of root growth ranges 65 to 87% of the controls. So this could be explained as the soil waterlogging has a direct effect to the root growth and causing the oxygen deficiency in the root zone. And another possible explanation could be the discontinuation of the seminal roots and decay of the seminal roots, which also causing the significant reduction of the root drivers under the water-flow condition. So plants started to grow thicker lateral roots instead of the seminal roots. So picture here shows that the decay of the seminal roots and developing new lateral roots under the water-flow condition. So there was a high shoot and root, high positive correlation between shoot and root growths, which showing that these newly formed lateral roots are becoming main functioning roots under the water-flow condition. So another notable observation was the roots altered state distribution. So roots distribution was altered among and above the soil surface, which is closer to the atmospheric oxygen. So all cultivars of subflowers, irrespective of the water-flow intolerance, started to grow surface roots. But visual observation that can be seen that those cultivars with higher water-flow intolerance, such as cultivar Meteora, subspecies Ienanicum, had grown more vigorous surface roots than these cultivars showing less water-flow intolerance, such as cultivar Rastel, subspecies Bradycarpa. So because of this newly developed lateral roots, the average root diameter increased on the well-drained condition. So it's also contributing to increased root porosity. So this is picture from the Gebert et al. 2001 that showing the increased root porosity after 35 days of waterlogging in Balanza clover, in subspecies Ienanicum, and in subspecies Saturnium. So root porosity is the gas-filled volume. In other words, it could be said that the percentage of gas space per unit tissue volume that is providing oxygen movement into the roots. So in this experiment, all cultivars of subflower increased the root porosity under the water-flow condition. However, the increased root porosity of subflower genotypes were below 10%, while the cultivar frontier porosity was beyond 10%, which is also could be explained its high water-flow intolerance. Another notable observation was the high constitutive porosity in subspecies Ienanicum. So constitutive porosity is a root porosity in a well-drained or aerated condition, which is also contributing internal aeration and water-flow intolerance. So in particularly cultivar Chickpea and Meteora had a constitutive porosity to beyond 3%. So we also considered that the increased root porosity is contributing for water-flow intolerance, which root porosity had a high positive correlation with both shoot drivets and shoot root drivets. So both leaf and petal growth were sensitive under the water-flow condition. So I measured every trifoliate leaf, combined 3 leaflets in every pot in every plant, and measured also the petal length in each leaves. So all subflower cultivars showed a significant difference in both leaf size and petal length, but the less reduction in petal length and leaf size in subspecies Ienanicum compared than the 2 other subflower subspecies is another indicator of its higher water-flow intolerance. So Also, the increased proportion of the leaf biomass above the ground enables the ionicum to increase oxygen uptake and facilitating root aeration and greater exchange of gases between plants and atmosphere throughout the increased number of the open stomata in the large leaves. So therefore, leaf size had a strong positive correlation with both shoot dry rates and shoot relative growth rates. Another point I would like to highlight here is that the 𝒂𝒓𝒊𝒂𝒂, even though 𝒂𝒓𝒊𝒂 has a post-rate growth habit, the inherently longer petals growth towards to the vertical position is likely to be another morphological feature of subspecies Aninicum to confer specific adaptation to the water logging. I also measured stomatal conductance. So stomatal conductance is a degree of the density, size, and opening of the stomata. So more open stomata, the greater stomatal conductance. So but in leaf stomata closure and the water logging can be caused the insufficient water uptake. So this is a phenomenon known as a physiological drought, which is water uptake into the roots is inhibited and causing insufficient water transport from the roots to shoots. So cultivar Denmark and cultivars of subspecies Aninicum maintained high stomatal conductance, which could be also explain the high tolerance. And this is also associated with the high root hydraulic conductance, high leaf water potential, and high transpirational rates. The stomatal conductance was the one of the key traits that had the strong positive correlation with both shoot and root growths under the water drought condition. I also assessed antecedent pigmentation in the petals. So all 𝒂𝒊𝒂𝒂 cultivars increased antecedent pigmentation under the water drought condition, particularly at the beginning stage of the water logging. And cultivars showing higher tolerance to the water logging gradually decreased antecedent pigmentation over the water logging period, which here the picture shows the two cultivars from subspecies Ceptrinum. The Denmark had the higher water logging tolerance, had less antecedent pigmentation than the Seton Park, which is showed less water logging tolerance, had the high antecedent pigmentation. So antecedent pigmentation is a water-soluble pigments that can be produced because both of the biotic and abiotic stresses, such as water stress. So therefore, fetal antecedent intensity can be used as an additional visual indicator of the strength of the water logging stress among the 𝒂𝒊𝒂𝒂 genotypes. So now I'm moving to the results under the recovery stage. So all plants started to recover after 21 days of recovery. So among the subspecies Ceptrinum shoot recovery was the highest, medium in brachycalicum, and the lowest was in subspecies Aninicum. So this could be explained as that the high growth under the water drought condition of subspecies Aninicum could be require extensive reserves such as carbohydrates and energy, which in consequence affected negatively of the shoot growth in the subsequent recovery stage. Another result was the root growth was the higher than the shoot growth. So which is plants prioritized root growth under the recovery stage. So this could be explained as the re-establishment of more optimal shoot to root ratio to maintain balance between transpiration and water uptake. And another notable response was the lateral roots and sulfus roots grown during the water logging were thinnest, and seminal roots started to grow back. So all other measured traits such as leaf size, petal length, stomatal conductance were recovered as the same levels as a well-trained condition. Only exception was the taproot recovery, So cultivars showed higher water-loving tolerance, such as cultivar Denmark and cultivars of subspecies Jiayin had greater taproot recovery. So they increased root depth is especially crucial as the flower growing in Mediterranean type climate, which is often suffer subsequent water stress during spring and summertime when rainfall declines. So the longer roots enables them to reach water available in the depth of the soil. So in the end, I would like to conclude that there were high variation in water-loving tolerance among the subflower subspecies, with superior tolerance in subspecies Jiayin, and there were water-loving were varied among the cultivars within subspecies, which is also suggesting that the future study should focus on the interspecies variation in water-loving tolerance. There were several key traits contributing to the water-loving tolerance, which were accumulation of greater proportion of leaf biomass, less reduction of stomatal conductance, higher constitutive and induced root porosity, and greater formation of new lateral roots. So this work has recently been published. So more measured traits and detailed discussion could be found from this paper. Thank you very much.
Any questions? Can I just ask, I wasn't certain about the experimental design, but what happens when there's a recovery, but if there's multiple waterlogging events? I think there was a taproot effect there. So if you had a waterlogging event, there's a recovery, but if there's another waterlogging event?
Might be.
Might be. It could be.
Roots,
Yeah. So
there's some water.
Does the recovery start to get less each time it's put under stress?
During the recovery stage?
Yeah, or so instead of just one waterlogging event and then we see the recovery in that species, is there a limit to how well a cultivar can recover from multiple waterlogging events?
I'm not sure.
I'm just because in the field I was thinking it's possible
fluctuating.
Any other?
I got two questions or clarification. One is you always showed the relative growth rate, which is okay, the way to analyze your data. The absolute dry matter of the one which is tolerant to waterlogging, I would like to see whether, to start with, they had a smaller dry matter. The relative growth rate is a good indicator of the waterlogging, but sometimes what happens in real world, the total dry matter will be still less. Was that the case? Of that waterlogging tolerant versus non-tolerant one? So the actual dry matter, the above ground dry matter.
After the recovery stage, the dry matter of subspecies Jiayin was lower than higher than the two other subflower subspecies, even though its growth was less, shoot growth was less, but the dry matter was still high. So that's why in the end I also would be conclude that in perspective of the dry matter, subspecies Jiayin is also doing the accumulating more biomass.
So in control conditions, what was the difference in dry matter? So take one which has the waterlogging tolerant, one which doesn't have.
In the dry matter, I also compared to the non-waterlogged condition.
Yeah, but were they higher in non-water?
Jiayin had the highest dry rates in control.
Yes. Okay. The second question is that the temperature you used is 20, 15 degrees, so 20 daytime and 15 at night. But in reality, I mean, Kevin and others may correct me. Generally, in the southern areas of Australia, West Australia, the waterlogging is earlier on, meaning at harsh winter time, June, and the temperature is generally low. But the temperature you gave is quite high. So my question is that if you take into a lower temperature, what will be the response? In fact, the waterlogging effect will be even
That's a really good question.
Anyway, let's share.
Yeah, the temperature is another affecting factor for the waterlogging. It's increasing the intensity of the waterlogging stress, I think. Yeah. So in this condition, of course, the glass house environment is fully provided everything there, and it was the environment which I agree with that. But the next experiment which I conducted last year, it was a little bit earlier than this time. This experiment was in July and August, September, something until to November, which is already getting warmer. But my last year experiment I started a bit earlier, but even though the growth was good. So it's not very high efficient, the effect I realized it from these differences. But of course, in the field situation, it's be very different, the cold situation.
I think probably there's no doubt that temperature in southern parts of Australia, during waterlogging velocity associated with cold soil temperature and cold air temperature, which would have an impact. So I suppose we could say that Gerelee is right, that is an artificial environment, but maybe the relative changes are the same across the species. But it would definitely be less dry matter produced in the field in winter. You probably see that. Thank you.
Also a prize for the last question, and you've been very patient.
That's okay. So my question about the recovery rate in the field when the clover is growing in a situation with other plant species, I'm talking about wheat mainly. So what will be the recovery rate compared to your controlled sort of environment experiments?
Recovery. Well, I when I started this experiment, I thought there would be a highly different recovery because related with the waterlogging tolerance. If they're behaving good under the waterlogged condition, they might be also to have ability more recovery, right? But in that situation, it showed the different results. So in the field condition, it's also might be very different because the waterlogging tolerance couldn't be assessed that how they're growing under the waterlogged condition. They might be growing very less in the waterlogged condition, but when the situation is improved and they no waterlogging, they could be grow vigorously because they had very less growth during the stress time, which they have more reserves to grow more vigorously. So in the field condition, maybe this after the waterlogging, they could be grow much vigorously after the waterlogged condition, and this could be also possible competing with the weeds. Does that make sense? Or I didn't answer maybe.
No, thank you.
You can discuss the beauty about the seminars is people get to know what you're doing, and so you can talk about that.
I just like lost my meanings after the end.
Now, Jiayin, you've got to promise me you'll never put pictures of food up at lunchtime. Never again. My stomach's been grumbling since 12 o'clock. So never again. But can we thank both speakers?